Today we're starting chapter 3 on macromolecules. Macromolecules are going to be larger molecules that are going to be made up of smaller things. So we're going to have 4 important different types of macromolecules that we're going to cover in this unit. So starting us out, we have this famous scientist here. This is Linus Pauling. He's the greatest, most accomplished scientist in history and known as the father of molecular biology. He's going to be born in 1901 in Portland, Oregon, and he's going to die in California in 1994. He was interested in chemistry at a very young age, so he went around and collected things, collected different chemicals and cleaning substances, and he performed or conducted different experiments at home when he was young. He graduated from Oregon State University with a chemical engineering degree in 1922 and received a doctorate in physical chemistry and mathematics from Caltech in 1925. He was a professor of chemistry at Caltech University of California in San Diego and at Stanford University, so he has a lot of accomplishments in several different places that he's taught. He studied and deciphered structures of biological molecules. That included things such as proteins and DNA, and he did a lot of this research at the same time of Watson and Crick and Rosalind Franklin, and so that was between the 1930s and 1950s. He does end up winning the Nobel Prize in 1954 and the Nobel Peace Prize in 1962. He established the Linus Pauling Institute of Science and Medicine in 1973. So he has a lot of accomplishments there. All right, so the carbon atom and macromolecules. Carbon atoms are going to be important atoms that are going to be used to construct a variety of biological molecules that are required by living organisms. It's a very important element. It does have 4 valence electrons, which is going to allow this atom to bond to various atoms, including things such as hydrogen, oxygen, and sulfur. So when we talk about things being living, lots of times we refer to it as being organic matter, and we could talk about organic chemistry. Those are going to be living things that contain matter. It's not the same sense that you would think of when we're talking about things such as organic plants or organic food. That's completely different. When we're talking organic matter, that means it's matter that came from living things, and it does contain carbon. Carbon skeletons are going to help carbon to bond itself and to produce like a carbon skeleton, providing a framework for building a biological molecule. The way that carbon bonds to other atoms, including itself, is going to result in a variety of different shapes and structures, such as long chains, it can make rings, and it can make branches. Thereby, this is going to help to provide living organisms with multiple ways to build biological molecules, which is going to be important. So it can make up many, many different things. It can make up millions of different molecules. So it's one of those elements that can bond and make up more molecules than most other elements can. And so here, looking at it on the periodic table, notice it's going to be in the 14th column there. Its atomic number is going to be 6. It has an atomic mass of 12. And so that means it has 6 protons. When it's stable, it has 6 electrons. And looking at that, since the atomic mass is going to be 12, you would do 12 minus the 6 protons, and that will tell you it has 6 neutrons. So organic molecules are going to be molecules that are composed of carbon and hydrogen atoms. Their functional groups are going to be groups of atoms that are going to be attached to that carbon skeleton, and it determines the chemical properties, the overall structure, and the function of an organic molecule. And so looking over to the side, you can see several of these macromolecules that are created. You have the hydroxyl, which has the oxygen in the center. It's got hydrogen off to the side, and then it has a variable unit. That's what's represented by the R, so that's where it can change. These are going to be polar molecules. You have a methyl molecule, a carbonyl, you have a carboxyl, an amino, a phosphate, and a sulfhydryl. So with these macromolecules, they're going to be large chain-like polymers that are going to be composed of numerous small repeating subunits called monomers, covalently bonded together. So poly means many, mono means one. So these polymers are going to be made up of these single units that are bonded together. It's very important in organic molecules because they provide living organisms with the starting material that's required to build cellular structures. There's going to be 4 main classes of macromolecules that we talked about. You're going to have your carbohydrates, your lipids, proteins, and your nucleic acids. Macromolecules are going to be composed of carbon, hydrogen, and oxygen, and they're going to exist in a 1:2:1 ratio. So that means you're going to have like that 1 carbon, 2 hydrogens, and 1 oxygen ratio there. So it's also going to provide a structural role for a variety of organisms, and it's going to help to play an important role in major energy source for sustaining life. So lots of these things we're going to take in, these macromolecules, we will take in for food. So 40% of our diet should come from the macromolecule carbohydrates. And so that's what they are made up of, mainly carbon, hydrogen, and oxygens. So some examples of carbohydrates are going to be your simple carbohydrates. You're going to have monosaccharides and disaccharides. So these could be things like sucrose. You could have complex carbohydrates, which are going to be your polysaccharides. These could be consistent of things like grains and breads. For simple, you could have apples and fruits. You could have sugary candies. Complex are going to be things that take longer to break down. Simple can be broken down very quickly. So with the simple, if it's a monosaccharide, it goes in and it will break that single sugar apart to release the energy, turns it into 2 pyruvates, and then breaks it down further to change it into energy molecules, releasing ATP. If it's a disaccharide, it breaks the bond between the 2 sugars that are hooked together and then starts breaking down that sugar molecule just the way it did with the monosaccharide. So these are things that can be broken down very quickly and used for quick energy. So think of your, like I said, your fruits, your sugars, your honeys, juices, things like that that you can get for quick energy. But those things are not going to be sustainable energy. Complex carbohydrates, the polysaccharides, are going to be many of these simple sugars hooked together. And so what has to happen is you have to break the bond between 2 sugars, and then you can break a sugar down. Break the bond again between it and another sugar, and then you can break that sugar down. So because these have many sugars hooked together, it's going to take a longer time to be able to break them down because you have to break the bond holding them together first, and then you can break the sugar. So these are going to be sustained energy. So this would be things such as pastas that you would eat or grains. And these would take time to break down. And so with these, they're not going to give you a quick burst of energy. They will give you long, sustained energy. So these are going to be the types of things that you would want to consume before you were to do let's say a marathon, or before you had a sport event. That way it continued to feed your muscles and continue to give you energy throughout that event that you're doing. Now, simple carbohydrates, like we said, consist of, let's say, 1 saccharide. A saccharide is a sugar molecule. Most common example is going to be glucose, and that's going to be a very important carbohydrate that provides energy to all living things. The reason that this is important is because it is the product of photosynthesis, and the sun is going to be our main source of energy. So this is going to be the simplest, most common way that we find sugars. Excess glucose can be stored in a human body or an animal body as a starch, and when it's stored, it's going to be stored as glycogen. That's the form of starch that animals store this energy as. So it gets stored in muscle or liver cells and then can be converted into triglycerides and stored in adipose tissue if you have enough glycogen storage already. So usually you have anywhere from 2 to 3 days of glycogen storage found in your muscle and found in your liver. If those are full, then it gets transferred and converted over into triglycerides, which are going to be fats, and then it's stored as adipose tissue, which is your fat tissue. And that can be stored around your body in different places. It could be in your tummy or belly. It can be found in your arms, your legs, wherever, and it can even be found around your organs as visceral fat. Other examples of these simple sugars or monosaccharides can be like fructose, galactose, deoxyribose, and ribose. Disaccharides. Di means 2, so these are going to be simple carbohydrates composed of 2 monosaccharide molecules that are going to form during dehydration synthesis reactions. And this is going to result in the formation of glycosidic covalent bonds. And so you will see that in that bottom picture there. So what happens is we're going to disassociate, or we're going to dehydrate, take that OH and then that H off, put those together, makes water. When you remove that water, it's going to hook that remaining oxygen together and hook those two molecules together. And so that's going to help to make your disaccharide. Common example of a disaccharide that most people come in contact with almost every day is going to be lactose. That's glucose and galactose put together through that dehydration synthesis. It's a very important carbohydrate because it is found in milk, and most people will eat products that contain milk or eat products that have had milk, mixed in with it. Other examples could include things such as sucrose and maltose. Some polysaccharides for carbohydrates are complex carbohydrates that are composed of multiple monosaccharide molecules that are bonded together through that dehydration synthesis reaction, just like what we saw in the disaccharides. These are going to function as energy storage and structural molecules. Common examples of these are going to be starches in plants. And plants store sugar as starch. Like I said, animals store sugar in an animal starch form, and that, remember, was glycogen. You can have glycogen, like we said, in animals, and cellulose in plants. Now, cellulose in plants cannot be broken down by humans. It can be broken down by some animals because they continue to chew. They have bacteria in their systems which help them break it down. But humans, we cannot break down cellulose, and we also cannot break down chitin. Chitin is going to be a carbohydrate found in things such as fungi, and also found in some animals that have an exoskeleton, so we cannot break that down. So cellulose and chitin are going to be indigestible carbohydrates for us. Now, they can be important in our food because they help to bulk up fecal matter, and that helps to push the food through our system and clean our intestines out. And that helps to prevent things like colon cancers. Now, lipids are going to be macromolecules that are also composed of carbon, hydrogen, and oxygen, but these are going to lack a defined composition ratio that we find in the carbohydrates. So due to their structural characteristics, they're going to also lack the ability to dissolve in water. So this makes them hydrophobic, or what's known as water-fearing molecules. So these are going to be things like your dressings, salad dressings, that you're not able to put together, things like Italian dressing that you have to shake up, but then they slowly start to separate again because of that oil or the lipids that are going to be found in them. They provide a variety of biological functions, include things like energy storage, which is a major function in our body. Major energy sources can be gotten from these storages in our body. So if, let's say, you are unable to get food for a day, or if you're in a famine, your body's able to break down these energy storages, and they're able to use these in order to feed your body and sustain yourself. They're going to be structural. We find them in places like the hypodermis, where you're able to help to insulate our body with lipids that are going to be found within. Also, we find vitamin composition and absorption important. So these are going to be for your fat-soluble vitamins, and protection and insulation, like I've already described. There's going to be 4 main types of lipids. You can have your triglycerides, your sterols, your phospholipids, and your waxes. First, starting off with the triglycerides. These are composed of a glycerol head that you see over to the side in that peachy orangish color, and the 3 fatty acid tails or chains. These include things such as saturated, unsaturated, and trans fats that you'll find in foods. So if it is a saturated fat, it's going to be characterized by straight fatty acid tails, which enable them to pack tightly together and to remain solid at room temperatures. So these are going to be things like the bacon grease that you may see solidify in a pan. These will be things like Crisco. These are going to be those things that are going to be solid at room temperature. Butter. Unsaturated fats are going to be characterized by the presence of little kinks in their fatty acid tails. This is called a cistail. It's going to have that little kink every so often, and that's because of that double hydrocarbon bond that you see there. This is going to make them less likely to pack tightly together, and they're able to remain liquid at room temperature. So these will be things like your vegetable oils, olive oils, fish oils, things such as that. So these will be the ones that are still able to be liquid. Triglyceride trans fats are going to result from hydrogenation. That's the addition of hydrogen atoms to unsaturated fats. That's going to change the structural conformation of unsaturated fats, and it results in a fat that's more closely resembled to saturated fats. This is going to be added when foods are going to be processed, and that helps to provide a more desirable taste and texture, as well as making it more shelf stable. So you have a shelf stable food at this point, and it tastes better and it gets a better texture. Problem is that it is not as healthy as the unsaturated fats. Sterols are going to be composed of 4 fused carbon skeletons with unique functional groups that are going to define their biological function. They're responsible for things like growth and development. Cholesterol is going to be a common example. They also play a variety of important biological roles, so integral components of cell membranes and structural precursors to steroid hormones, things that our bodies need, such as estrogen and testosterone. Phospholipids are going to be composed of a glycerol head and 2 fatty acid chains with a phosphate group. Now with this is what you're going to see in things such as cell membranes. So these are going to compose with hydrophobic and hydrophilic properties. So the head, which you see there, is going to be a hydrophilic head, and you see it has a phosphate glycerol component. That's going to face towards the inside of the cell because it has a liquid environment with its cytoplasm, and it's also going to face the outside portion of the cell where there's usually a liquid environment in that interstitial fluid. The fatty acid tails will then face towards each other. In between the bio layer or the double layer there, it's gonna not have any type of liquid there, so that's where they're happy. If fluids do get in there, then they kind of scrunch up and try to get away from that fluid. So like we said, main structural component of a cell membrane, and you can see here with that bilayer how the heads are going to face out and inwards. And they're arranging themselves in this way because they are reacting to the watery environment from the inside of the cell where the cytoplasm is, and the outside of the cell where you're going to have that interstitial fluid. So that glycerol head with the bonded phosphate group is hydrophilic, which means it's water-loving and faces outward towards either the inside of the cell or the outside of the cell. The fatty acid tails are hydrophobic, which means water-fearing, and they're going to be oriented inward towards each other. They make a bilayer nature of the cell membrane, which enables it to regulate the movement of substances into and out of the cell. So it kind of makes them like a gatekeeper. They will determine what can come in and out. Waxes are going to be composed of 1 fatty acid chain, a glycerol head, and a long chain alcohol. A variety of characteristics, including high melting points, existing as solids at room temperature, and being hydrophobic, are going to be common things that you see with these. They're found in protective coatings on plant leaves and stems, and are going to be responsible for maintaining animal skin and fur. Our next macromolecule is going to be proteins. Proteins are going to be composed of amino acids that are bonded together by peptide bonds and formed from dehydration synthesis reactions. So biological functions of these are going to be things such as enzymes. Enzymes are going to allow chemical reactions to occur at, let's say, lower temperatures or with less reactive energy that's needed. Proteins are also important in defense. They're important in transport. They help bring things into and out of a cell. They can take things through the cell, attach to them and take them through, or even attach and bring things through the body. They also help with regulation. They can be at the cell membrane, and they can regulate specific things that are allowed in or specific things that are allowed out. Proteins are responsible for structural functions. We have proteins making up our hair, our skin, our fingernails, and our muscles. All things in our body can be made up of different types of proteins. They're responsible for mechanical functions. Think of your muscles, moving things around. Responsible for movement and responsible for storage. Like we said, proteins are made up of smaller units called amino acids. These molecules that form proteins are going to be made up mainly of 20 different types of amino acids, and they're going to be in combination of these amino acids which provide function and structure of proteins. Now, all amino acids contain the same basic molecule. That includes an alpha carbon, to which is bonded a hydrogen atom, an amino group, a carboxyl group, and an R group. Now, the R group is going to be important because that distinguishes the different type of amino acids. The other parts, the amino group and carboxyl group, remain the same. So the R group is going to change. And so how a protein folds into its unique three-dimensional configuration is going to be dependent upon this. And that's going to determine the function of the protein. So if a protein unfolds or if it doesn't fold properly, then it won't function properly. Examples of amino acids you see here. And the protein structure and primary structure is going to consist of a simple linear sequence of amino acids. This one here is insulin. And so it puts together amino acid 1, 2, 3, 4, and so on. You see 11 hooked together here. And so what happens is DNA is going to be read, and it's going to take 3 codons at a time. So let's say A, T, C. It would take those, it's going to find the matching amino acid, it brings it to the ribosome and starts hooking them together using peptide bonds. And so these little structures that you see in between each amino acid is a peptide bond. So amino acids hooked together by peptide bonds can be called a polypeptide chain. Now, when you're hooking them together, the first structure of a protein is called this here that you see, the primary structure. It's a linear line of amino acids hooked together with peptide bonds. Next, you go into what's called the secondary structure. What happens here is it takes the amino acid chain or polypeptide chain, and it's going to begin to fold them. Now, it can fold them in a corkscrew fashion or into almost like a zigzag fashion. So you have 2 different types produced by how the amino acid folds. So you can have what's known as an A helix or a B pleated sheet. So A helix is what you see below on the left-hand side. The B pleated sheet is what you see on the right-hand side. And these are going to be maintained by hydrogen bonds. So the A alpha helix that you see here below is an example of keratin, and the B pleated sheet that you see over to the right-hand side is going to be silk. Now, the third structure that it has to be folded into is going to be the tertiary structure. And so what happens here is it folds it, and because of how it folds it is going to be how it's interacting with water. So you can see myoglobin. And then the quaternary structure is when you take these structures that you see here at the tertiary structure and you start to hook them together. Once you hook them together, it could be 2 or more small tertiary protein structures hooked together. That's when you have a fully formed protein. So here, like we said, hemoglobin, it's got 4 of these tertiary structures hooked together. 2 alphas, 2 betas. And that makes your hemoglobin. So no, proteins have to go through 4 different structures. First, you put them together, the primary structure. Second, you either fold them or zigzag them. That's the secondary structure. Third, you bend them and you use water to kind of bend them away to make this 3-dimensional structure here. That's the tertiary. And then you hook these tertiary structures together to make a functional protein, and that is the quaternary structure. Proteins only work when they're in the correct shape. You can do a process called denaturation to denature a protein, and that's a protein whose structural interaction has been disrupted by means of various environmental factors. Now, pH can do this, temperature can do this, even mechanical things can do this. And so that causes that protein to unfold and to lose its overall function. So here you see a proper functioning protein to the left. You added heat, it unfolded it, and you see its unfunctional shape over to the side. So it lost its overall function. Shape equals function in proteins. So if it loses its shape, it loses its function. You can do this mechanically. You can take egg whites and you can take a beater and you can beat them And once you do that, you break these bonds, you unfold them, and you can make meringue with them. And so add a little bit of sugar. It's nice, sweet meringue. You put it on top of a pie. But you changed it from that clear egg white into that fluffy white stuff that you put on top of pie by denaturing that protein. Enzymes and activation energy. Proteins that are responsible for speeding up chemical reactions by lowering the activation energy needed. Activation energy is the required energy to even start a chemical reaction, so you can lower how much is needed by adding an enzyme in. Enzyme activity has a 4-step process. So first, you have the formation of the enzyme substrate complex. A substrate fits into the active site of an enzyme. It's explained using 2 models here. It's like a lock and key system. So you see the enzyme is going to be like the lock, and the substrates are going to come in together, and they fit in kind of like a key. So as long as the enzyme has the proper shape, that key fits in properly. But if something happens and it changes that enzyme, it doesn't fit in properly, and therefore it can't break that substance apart any longer, or it doesn't work properly. So enzyme activity, the second step, that transition state, the substrate exists as an intermediate form. Third, the product formation, the substrate is going to be converted to a final product. And then fourth, that product release. So it releases it at the active site. Factors affecting enzyme activity. Well, you can have enzymatic activity being affected by a variety of chemical and physical factors, including enzyme substrate concentrations, temperature, and pH again. So these can affect how those enzymes work. Enzymatic activity is also going to be affected by activators and inhibitors. So example, snake venom is going to be a competitive inhibitor, while cigarette smoke is going to be a non-competitive inhibitor. So factors that could affect this rate, we here see that substrate concentration. You can have a non-competitive inhibitor. Notice how its rate of reaction it enhances and then stays constant. If it's a competitive inhibitor, it rises and continues, but it rises at a steady rate. And then you have normal enzymes, which will come in, and they're usually at higher rates. So macromolecules are going to be composed of nucleotides for nucleic acids. And so with these, those most that you know of are going to be your A's, T's, C's, and G's, and that's going to be for DNA. So DNA is made up of nucleic acids and nucleotides. You can also have uracil, which is going to be found in RNA. So that would be a nucleotide found in RNA. So nucleotides are going to have 3 main components. They're going to have a phosphate group, they're going to have a 5-carbon sugar, and a nitrogenous base. And when we talk about the A's, T's, C's, G's, and U's, those are the nitrogenous bases. Deoxyribonucleic acid is going to be composed of a double strand of nucleotides that are going to be twisted into a helix. Deoxyribose is going to be the sugar that's found in the backbone. Nucleotides of DNA are going to consist of a phosphate group, a 5-carbon sugar, which we just said was the deoxyribose, and 4 nitrogenous bases. So it could be adenine, guanine, cytosine, and thymine. Adenine and thymine always go together, and cytosine and guanine always fit together. Nitrogenous bases are going to be complementary, and that's what I mean by when they fit together. So those base pairs with adenine bonding with thymine and guanine bonding with cytosine. And notice how your thymine and adenine, when they bond together, they make the double hydrogen bond there. And when the guanine and cytosine bond together, they make that triple hydrogen bond there. It's going to be found within the nucleus of the cell, so that's where we find DNA, and it's going to outline your genetic makeup of that organism. So it helps to provide the cell with necessary instructions for building different types of proteins. And so this is where you're going to have your mRNA come copy a gene that needs to be produced, and it's going to take it to the RNA, like I described in the structure when we were putting that polypeptide chain together. Ribonucleic acid is going to be your RNA. That's composed of a single strand of nucleotides, which consist of a phosphate group, a 5-carbon sugar, but in this case it's going to be ribose instead of deoxyribose, and 4 nitrogenous bases. So you have adenine, guanine, cytosine, and this time uracil instead of thymine. RNA is going to be found outside of the cell or outside of the nucleus of the cell. So it can come in, it can make an mRNA strand, but it's going to exit outside of the nucleus and go into the cytoplasm in order to build itself. So it's responsible for translating DNA genetic codes to make proteins. Structural comparisons that we can see here. So with the RNA, you're going to have one strand. You have C, G, A, and U. With DNA, you're going to have a double strand, and you're going to have C, G, A, and T. Also, RNA is going to be made of ribose sugar. DNA is going to be made of deoxyribose sugar. That's going to end our unit on macromolecules. I'll see you back with our next unit.